What is cystic fibrosis? Cystic fibrosis is a rare genetic disorder caused by a mutation in a gene called cystic fibrosis transmembrane conductance regulator. The CFTR gene controls the flow of salt and fluids in and out of cells. Cystic fibrosis also causes abnormal production of thick mucus and blocked airways. Patients with cystic fibrosis have a high chloride concentration in their sweat. The following are the common symptoms of cystic fibrosis: persistent cough that produces sputum, poor weight gain and growth, greasy and foul-smelling stools, as well as a very salty tasting skin. Sweat chloride testing is the gold standard for the confirmatory test of cystic fibrosis. It measures the conductivity and amount of chloride concentration in the sweat sample. One of the quantitative pillocarbine iontophoresis test methods is the 1983 Wesco macroduct sweat test system, which is a continuous flow principle. The following are the steps on how pillocarbine iontophoresis works. Firstly, topical pillocarbine propelled transdermally by electromotive forces using a weak electric current which is 1.5 to 4 mA for 5 minutes. Then, pillocarbine stimulates muscarinic receptors on the eccrine sweat gland that will induce the secretion of sweat. Why use pillocarbine? This is because pillocarbine is used to stimulate sweat glands in a sweat test to measure the concentration of chloride and sodium that is excreted in sweat. For the test preparation, no special diet, no activity limit is needed. Patients may eat, drink, and exercise as usual before having this test. It is important for patients to be well hydrated before undergoing the procedure. Any regular medications can be taken as this will not affecting the test results. Next, patients should be instructed to avoid applying any creams or lotions to the skin 24 hours before the test is done. So as for the test sample, we need a sweat sample collected using pillocarbine iontophoresis method. The sweat must be collected from arms or legs only to prevent the iontophoresis from stimulating the heart. The sweat must be collected in a macroduct coil using techniques to minimize evaporation and contamination. This procedure involves 3 parts, which is sweat induction, sweat collection, and sweat analysis. For sweat induction, first, clean the selected skin area, then insert a pillocarbine gel disc into both electrode and attach the electrodes to the limb. A weak electric current will activate the iontophoresis. For sweat collection, after the iontophoresis is complete, remove the electrodes and clean the stimulated skin. Then, place the macroduct sweat collector over the stimulated skin site and strap in place firmly. Collect the sweat for 30 minutes. After approximately 50 microliters of sample is collected, remove the protective transparent cover and attach a blunt syringe to the open end of microbe tubing to uncoil it completely from the collector. For sweat analysis, syringe is used to pass the sweat from the microbe tube into sweat check analyzer. This analyzer will measure sweat conductivity, calculate electrolyte concentration, and display the reading in equivalent sodium chloride molarity units. The normal range of sweat chloride test for infants should be less than 29 mmol per liter, while children and adults should be between 10 to 30 mmol per liter. Test may be repeated if chloride level for infants is between 30 to 70 mmol per liter and chloride level for children and adults is between 40 to 70 mmol per liter. But if the chloride level is above 90 mmol per liter, it is likely to have cystic fibrosis. Next, we will go to the condition that can cause false positive and false negative of sweat chloride test. We characterize into 2 conditions, which is because of technical error and diseases. For false positive results in technical error, it is because of errors in measurement of sweat chloride concentration, such as evaporation of sweat sample and erroneous dilution and calibration methods. Meanwhile, anorexia nervosa and atopic eczema are disease conditions that can lead to false positive results. For false negative result in technical errors, it is also because of dilution of sweat samples and inadequate sweat collection. In disease condition, false negative result can occur because of ectodermal dysplasia and edema diseases. Lastly, advantages of this test is it is more reliable in diagnosing cystic fibrosis and it is unthreatened to the infant since it is needle-free test. For this advantage, the test needs sufficient amount of sweat samples to prevent erroneous or repeated tests.